Okay, well, today marks a turning point in the course. We are on the kind of last section. We'll be talking about communities from here on out, communities and ecosystems. So this is the big picture. Um, and we will cover 4 topics today. Now, it's important to realize that the hope way back in the mid 1970s was that we could do what we have just done with pairwise interactions, that is, competition, predation, and mutualism, and generate something like logistic equations for each of those interactions so that we could just add a term to the logistic part, the part that accounts for interactions. And so we would be able to basically write an equation for a community. So dn dt equals r times n times k minus n over k. And then add in those terms for competition and predation and mutualism to that k minus n over k. But we can't do that because those coefficients aren't stable. They change as you add or subtract species. So we had to give up on that. So the kinds of questions we ask of communities then don't arise directly from the kinds of information we can get from populations directly, and they don't arise directly from pairwise interactions. So we kind of carve out pairwise interactions and analyze them separately. We analyze communities on their own terms. So we'll focus on things like diversity, like succession, which is the patterns of diversity over time that a community exhibits. We'll look at community structure. Which species are really influential in maintaining the diversity of a community. But we are not going to build directly on information that we've been working through and when we're talking about individual populations or pairwise interactions. That was the great hope. It didn't turn out to be true. So that's a really good example of how emergent properties force us to adopt different means of analysis. So communities don't rely nearly as much on equations or on those graphical things we've been doing. They are on their own terms. And they are much more conceptual. So we will be talking about different kinds of stuff from here to the end of the course. So first we'll just talk about what we conceive to be ecological communities. We'll talk about indicator species and their uses and abuses. We'll spend some time on whether communities are open versus closed. And then we will start talking about the single most important aspect of community ecology, which is diversity, and how we measure diversity. Those of you who are in the lab already know something about this, but we will make it operational for everybody in the class now. So, how do we think of communities? Well, we think of communities as an association of interacting species that co-occur in time and space. They have to be physically together in some sense in order for those interactions to be meaningful. And we frequently refer to communities by using what we call an indicator species. Now, you have to be careful when you're talking about indicator species because it's easy to attribute more importance to an indicator species than is actually warranted. This is just really basically a convenience for us as ecologists, so that we kind of know what we're all talking about. So an indicator species reliably and consistency predicts the presence of other species. But it's not like that indicator species has special significance. It's just like at Jazz Fest where they put up a flag and everybody gathers at the flag. That's kind of it. It's not like that flag has a special significance just because it has, 3 flags on it that everybody recognizes. Well, the importance of that is that everybody recognizes those 3 flags. It's not that they have some special significance. It's not like if there's a pirate flag on there, that there's actually going to be pirates there. That's not what it's about. So a real good example around here is longleaf pine savanna. And that is named for the largest and most obvious plant in the community. Okay, it could just as well be named for the, say, Sarracenia insect-eating plants that occur in these wet communities. But we use the largest and most obvious plant, not the tiny little really significant thing that occurs nowhere else on Earth. So when we talk about longleaf pine communities, everybody sort of knows we're talking about a wet savanna that has a lot of carnivorous plants, that has a really high diversity of flowering plants in the community, and that you can usually, guarantee is going to be there if you see longleaf pines. Okay, it's usually a large or numerous species, but it's not really the most important species. It's really crucial for you to understand that. It's not the most important species. It's just like a flag at Jazz Fest. Everybody knows what it is, where it is, and can talk reasonably about the same sort of thing. So longleaf pines are obvious, but it's not like they structure the community. It's not like they're structuring the environment so that all these carnivorous plants can live underneath them, even though that's what the pattern in nature is. It's just an indicator. And we can talk about longleaf pine communities and know what each other is talking about. Now, here's an important thing about indicator species, right? They reliably and consistently indicate the presence of other species. But does that mean that the composition of a community is always consistent? Okay, well, that's a different matter. Here's how we might think about these things. If you have a closed community, then an indicator species might actually indicate a consistent community composition. A closed community is a discrete unit with sharp boundaries, and we call those boundaries ecotones. That's an area of rapid replacement of species along some gradient. Okay. So you might be able to walk over, say, 20 or 50 meters and move from one community to another community. And a bunch of species will turn over. The species that you started in will all drop out, more or less simultaneously, and be replaced by a whole bunch of other species. That's what an ecotone indicates. It's a sharp gradient in species replacement. So what that would indicate is that there are probably really strong interspecific interactions, and that each species plays a specific important role in the community. If you lose one of those species, because interactions are strong, you probably are going to lose most of them. And the analogy to a closed community is like a superorganism. Like each species in the community is playing some role analogous to the organ systems within an organism. Okay? On the other hand, you could have open communities, that are nothing like this. So in an open community, you have vague and distinct overlapping boundaries. There's no sharp ecotone here. What you got is a bunch of individual species distributions that are kind of following their own their own niches, right? And their own sets of requirements. Well, there's no ecotone here. This is just a bunch of species that happen to be co-occurring. And what that indicates is that species distributions are determined by physical factors, not biotic interactions. Here, you might make a reasonable case that the interactions among these three species are important. And if one of them is missing, the other two probably are not surviving. Here you have a set of four species that tend to co-occur, and you might imply that this might imply that these four species interact strongly, that their co-occurrence is really important. Down here, they might co-occur, but it's because they're responding to physical features of the environment that determine their physical niche. So what this implies is strong biotic interactions. This implies that there are weak biotic interactions, and that species could easily substitute for one another here, whereas they can't here. So the role of an indicator species in these two kinds of communities is quite different. An indicator species in this kind of situation, where you have closed communities, really does indicate that something important is going on with these interactions. An indicator species down here, let's say this one here, that just indicates, hey, this is someplace on an environmental gradient where you typically find these species. It doesn't imply anything about how important those interactions are. It's just the classic convenience indicator. Here, an indicator species might actually mean something important in terms of biotic interactions. Here, it just is a sign, like, we're here on this gradient, you can expect to find these other species here, because they're responding more or less to the same things on the gradient. So what do we find in nature? Do we find a lot of closed communities, or do we find a lot of open communities? Well, as it turns out, most communities in nature appear to be open. So here's a really good example. This is an open community in space, and these are open communities in time. We'll talk about both. So in the mid-20th century, people defined something called a cove forest community of the Smoky Mountains. Now, this was a set of tree species that you could reliably find in these little valleys, mostly on the western side of the Appalachian Mountains. And you've always found a set of about a dozen species in these little valleys. They were cool, they were moist, they were west-facing, so they got afternoon sun a lot. And the idea was initially that these cove forest communities were something special, like, the species in them were probably interacting. Well, then we started doing species distribution maps, and we realized, that's not what's going on at all. This just, whoops, sorry about that. I thought I turned that off. Actually, I did turn it off. Why is it beeping? This is just a situation in which the distributions of these dozen or so species happen to overlap on the western side of the Appalachian Mountains in these valleys. There's nothing special about them because, look, some of them have their southern boundaries right here and their northern boundaries in Canada. Some of them have their northern boundaries right here and their southern boundaries along the Gulf Coast. Well, there's no way that there's anything special going on here. This is not a unit of interacting species. It's just a situation in which species happen to co-occur because their physical niches happen to be satisfied in these valleys on the western side of the Appalachian Mountains. So this is an open community. We can define it for our own convenience. We can call it the cove forest community of the Appalachians, And everybody will know what species we're talking about. But it's a mistake to attribute more to that co-occurrence of species than is actually the case. These species, yeah, they co-occur, but it's not like they're strongly interacting. Now, we can also look at the co-occurrence of species in time. So what this is here is the record of post-glacial dispersal of trees from different refugia. So during the last ice age, glaciers sat over the Great Lakes and were actually down into Ohio, into New York. Long Island in New York is a glacial moraine. It's a pile of rubble that the glaciers pushed south and deposited at the end of the glacier. So all of this area in North America and, say, the northeastern part of North America was under 2 miles of ice. Well, there's no community there, right? Well, what we find today is a very distinctive deciduous forest community. It's the deciduous forest of eastern North America. But, as it turns out, we can trace the dispersal of those species from different refugia, where they sat for about 40,000 years while ice occupied most of North America here. So there was one refugium on the Gulf Coast, and there was another one in the Carolina coast. Okay, and the species that lived on the Gulf Coast while the ice was there had absolutely no contact with the species that were in the Carolinas. So for roughly 30,000 years, this species here, maple, which is currently considered part of the Great Eastern Deciduous Forest of North America, had absolutely no contact with a whole set of other species, which is exemplified by hemlock here. Hemlock was on the Atlantic coast in the Carolinas and had absolutely no interactions with maple, which was stuck on the Gulf Coast. So the fact that they co-occur now in the Great Eastern Forest of North America is a complete accident. They dispersed at different times, at different rates, from their refugia. So this tells you how many thousands of years ago we have traces of pollen from that species being present in lakes that occur in these areas. So you can see that roughly 12,000 years ago, we had hemlock pollen in the Carolinas, and we had maple pollen as long as 18,000 years ago in the Gulf Coast. But there was no maples 18,000 years ago in the Carolinas. That did not happen. And you can see that they dispersed at different rates. Hemlock reached the southern Great Lakes by about 10,000 years ago. And maple did also, but look, maple also didn't reach some of these areas in the Carolinas until about 12,000 years ago, by which time hemlock had started dispersing north quite rapidly. And although hemlock occurs in, say, Wisconsin today, it didn't get there until about 6,000 years ago, at which point maple had been there for at least 1,000 years. So it's not like these species are interacting. They're completely independent of one another. And they occur in what we call a community today, simply because that community satisfies their physical niches, not because this is some kind of superorganism. This is pretty typical of what we find in nature. We don't find closed communities all that often where there's a sharp discontinuity in species composition. Where do we find closed communities and changeovers in ecotones? Where do you think? It's where biomes abut one another. So the eastern forest biome, where it changes over to the prairie biome. Okay, and at that point, you get a sharp discontinuity where all of the trees in the eastern deciduous forest drop out over a space of about 50 to 100 miles and are completely lost. At which point, grasses take over. And similarly, if you're moving from west to east across that environmental gradient, you lose all of the grasses almost immediately. And all of the associated species in the grassland biome are lost within about 50 miles. So it's where biomes are abutting one another that you see sharp ecotones. But you see relatively vague and indistinct ecotones, say, within the deciduous forest or within the prairie communities. And a really good example that's kind of easy to remember is that aquatic biomes, like lakes, are embedded in forest biomes. Okay, well, the forest species don't go into the open water, and the aquatic species don't go onto dry land. There's a really sharp ecotone there. That's a really good example of where two biomes are close together over a very short space, and you find a sharp ecotone. But most communities in nature, we think, are open in terms of both time and space. So this is a subject where it's actually really important to try to define what the species turnover is like. So we spend a lot of time on this when we talk about community ecology. And I have a little example problem for you. So if you'll get your clickers out, let's have a look at kind of a real problem that we face when we're thinking about ecotones and open communities and closed communities. And this will illustrate how the physical niches of species might influence your thinking about whether you have an open or a closed community. So here we go. This is actually not, it is a very simplified example of something that we deal with right here in Louisiana. So here is the frequency of riverine fishes on a salinity gradient, and here's the frequency of estuarine fishes on a salinity gradient. And so here you see that the river fishes are pretty happy in low salinities, and here's their distributions, and you'll notice where they all have their limits. Well, now here are the estuarine fishes that you might find in Lake Pontchartrain, right? And you can see that they all prosper in much more saline environments. And here's the range at which they drop out as you get fresher and fresher and fresher water. So from here to here, these are the estuarine limits. Here's the limits of the freshwater river species from here to here. So how does this match? What do you think would most closely match the way that these two fish communities are organized? Do you think that they're basically an open model? Is it because of the wide overlap? Is it an open model because there's no visible ecotone? Or is that wrong because that's an ecotone? Is that closed because there's an ecotone for the river community? Is it closed because of the high overlap? Or is it a mixed model? Is it more closed for river fish and more open for estuarine fish? So this is going to not allow us to do anything for just a minute. Okay, but now it says it's open. So let's see what you think. 5, 4, 3, 2, 1, and done. And let's see what we can do here. So we have quite a mix of opinions here. All right, well, why don't you talk it over and argue about it and revote. So the first question, is it open, closed, or mixed? The second question is, why is it open or closed? Because it could be because of the overlap or because of ecotones. Okay, let's revote and see if we have movement in kind of the right direction. But this is quite typical of the kinds of things that we have to look at when we're trying to understand communities. Do we see sharp boundaries? Do we see indistinct boundaries? What does that tell us about the relative importance of biological interactions versus the physical niche? And 5, 4, 3, 2, 1. Okay. Okay, so most of you are now going for a mixed model. And let's have a look at that. Let's see what the rationale is. So what we're looking at here is a relatively tight boundary here for low salinity, which you kind of would expect if it's a freshwater fish, because really they should be able to tolerate pretty low salinities on this end. But notice that they're all dropping out in a relatively short range here, kind of in the middle. The range for the estuarine fish is much larger, from here to here. So it's not the same. There is kind of an ecotone, sorta, but it's messed up. It's messed up very specifically by the broad tolerances of the estuarine fish from here to here. It would be really legitimate to talk about this as kind of a mixed model, where the biotic interactions are more important for the riverine fish, and maybe their environmental tolerances here are really more sharply drawn than for the estuarine fish. The estuarine fish have a much broader range of tolerance of salinity. So it's a bit more open for the estuarine fish than it is for the river fish. Okay, this gives you some clues about how important the physical environment actually is. Looks like the physical environment is somewhat more important for river fish than it is for estuarine fish. Okay, and this is an actual simplification of an actual species distribution thing. So it's exactly the kind of thing that we always ask about in community ecology. Okay? So questions about thinking about open versus closed communities. All right. Well, let's talk about species diversity. How do we understand species diversity? This is the single most important question in community ecology. How many species are there, and why do communities differ in the number of species that they have? We will spend a couple of lectures on this, working at different scales, on the local scale and the global scale. But what we need to do today is just say, how do we measure this? So there are a couple of ways in which we commonly do this. First is just to count the number of species, right? So here your diversity is 5. And here your diversity is 5 also. Now we call that species richness. Richness is simply the number of species in a community by convention in ecology. But clearly these two communities are not the same, because you have only 4 individuals of these 4 species and everybody else is species 1. And here you have much greater evenness. Each species is represented by 5 individuals. So they have the same richness, 5 species, but very different evenness. Now this is important, and it guides our decision making in a lot of cases, especially in conservation models. Because a community that has lots of individuals of a species of particular concern, like this one, might be a target for conservation if your target is simply this species. But if you are interested in diversity, like a wide representation of a lot of species, this is a crappy community to conserve. This one is much better for overall diversity. Now, how do you think about diversity? How can we think about it in a way that makes sense? Imagine yourself standing in the middle of a random point, standing at a random point in the middle of community 1. We have 21, 1, 1, 1, 1. Look around you. What are you likely to see? Species 1. You're not likely to see species 2, 3, 4, or 5. That's kind of a way to visualize how that community is organized. If you're standing at a random point in the middle of community 2, what can you expect to see? You're much more likely to see all of those species. So there's a real sense in which community 2 is more diverse than community 1, even though it has the same richness. So we have many, many, many measures in ecology that try to take this into account. There are so many of them that there are 300-page books written about just measuring diversity. We're not going to deal with this. We're going to deal with the single most common way in which we contrast richness and evenness. And that's the Shannon-Weiner index. So it's clear that community 2 is more diverse than community 1 in a very meaningful way. Same richness, but if we take evenness into account, community 2 beats community 1. So we need an index. We need some way of comparing them quantitatively, and here it is. And you'll get this for homework. Some practice in calculating this. This looks ugly, but it's actually not. If you have a calculator, you just need to know how to read the equation here. The Shannon-Weiner index is an information index. It was invented in the 1940s by Bell Labs to quantify the amount of information that could be sent over a phone line of a particular type. So they could compare the efficiency of information transfer across phone lines. Well, it turns out that it's not a bad way to think about diversity because it adjusts the richness, which is the proportion here of each species, by the evenness of that species. Its relative abundance, which is this part here, the log of pi. So H prime is your value of Shannon-Wiener index, also sometimes called Weaver. And Shannon and Wiener were the Bell Lab scientists who invented it. I forget what Weaver did. Weaver had something to do with this. But H prime is the total. So what you do is you multiply the proportion of each species by the log of the proportion. And you do that for species 1. And then you find the proportion of species 2 and multiply it by the log of species 2, proportion of species 2. And then add that in. Now, the log of a proportion is always a negative number, right? The log of something less than 1 is always a negative. So, you just change the sign. And I see that we start with species 1 here and we go to however many species there are in the community. But this is the proportion of each species times the log of each species, and what you do is just use your calculator, and this is how you work through it. So the proportion of this species in community 1 is 0.84, and the proportion of each of these is 0.04, and notice that they total 1. Got to make sure you're doing that right. So the log to base E of this is minus 0.174. So pi times log pi is 0.84 of this, 0.146. The log of 0.04 is a relatively large negative number, right? But this, the proportion, times the log of the proportion is that. Okay, so you add these things up, and this is what you get. This is your total diversity. Okay, and all you're going to do is change the sign here. So it's 0.662. All right, so for this one where it's all even, fortunately, you only have to do one calculation. Because the proportion is 0.2 for everybody, and the log to base E of that proportion is 1.609, or negative 1.609. So this times this is this, and then you add them all up, all 5, and you get minus 1.61, and so you change the sign. So now you have a way to compare. This is much greater than this, which accounts for the greater evenness. So it's richness adjusted for evenness. And we frequently will make comparisons of communities on the basis of this very simple index. It does have some undesirable qualities because it asymptotes at about 4. So even if you have 1,000 species and they're all equally common, you're still going to get a diversity of about 4. But if you have one species in the community and only one, the log of 1 is what? 0. Yes, thank you. One of you remembered your algebra. The log of 1 is 0, so anything times 0 is 0. So the diversity of a community with one species is by definition 0. So it ranges from 0 to about 4.5 something. And it is a very useful way of comparing communities. This is probably the single most common index that we use in ecology. There are a number of others, though, and given our fondness for graphs as ways of trying to sort of visualize complicated information, shouldn't be surprised that we use graphical approaches also. So we use something called the rank abundance curve very frequently if we're not satisfied with the Shannon index. And this is what a rank abundance curve looks like. So here's those two communities. So here's community one, community A, and that had 21 out of 25 individuals in this species. And here's the other 4, right? And here's 0.2, right? It's on a log scale, so this is 0.2. So this is your second community, community B. And look, you can get a relatively rich set of information out of this graph without hurting your brain trying to digest a large column of numbers. So community A had 21, 1, 1, 1, 1. And look, the most abundant species is right here. And the slope here is really sharp. This is a really high slope. Well, what that tells you is that the change in evenness from, the change in abundance from the most abundant to the next most abundant species is really high. Well, that tells you that the most abundant species are really dominant. The low slope here indicates that most of these species are about the same abundance in the colony, in the community. Notice that if you have more than 5 species, that tells you about the richness. So this axis tells you about richness here. The longer the tail of your distribution, the more species are in the community. The higher the slope, the lower the evenness. So this is a good way of just getting a snapshot of a community and being able to quickly figure out the relative abundance and evenness of the species in it. So the steep slope here means that you have real dominance by one or a few species. A shallow slope means that they're pretty even. So let's look at a couple of real communities. And here's what we have. Okay, well, look, here's the red line. This is the Gulf of California. This is the diversity of species of fishes in the Gulf of California. And look, they start at about the same place with the most abundant species, but which drops faster? The blue line for the central Gulf of California or the red line for the northern Gulf? It's obvious. The northern Gulf of California drops much faster. The relative abundance of these species is much lower. So there's greater evenness in the central Gulf of California than there is in the northern Gulf of California. And that's because the Colorado River empties into the Gulf in the north. And so these species have to be tolerant of both a higher range of salinities than these guys. This is a more benign environment for marine fish. Also, look, there are 53 species in the northern Gulf, and there are 57 species in the central Gulf. Richness, and the slope is evenness. Okay, many species are much, much, much more distinctive than this. And here's a really good example. So here's the richness and evenness of insects in mountain streams versus coastal ponds. And you see that coastal ponds are dominated by one species. Okay, we're not specifying which one it is. That's a different question. But look, there's very few species in these ponds. There's only about 18 species of insects. Richness is lower. And you can see from the slope that the evenness is very unequal. One of the species is really dominating this environment. In mountain streams, look, the most dominant species only accounts for about 20% of the individuals. Remember, it's a log scale. And there are many, many, many more species here because it's a more complex environment. So you get as many as 76 or so species in the mountain stream, and you only get 19 or 18 or 19 in a coastal pond. Well, it's really easy to get this information off the graph. I mean, the slope tells you instantly how even they are. Pretty even, pretty uneven. Real dominance here of one species. And the number of species is read directly off the x-axis. So richness differs greatly, and evenness differs greatly. Different slopes, different endpoints of the distributions. Okay. Questions? When we talk about diversity, try to keep these methods in mind. Try to keep the Shannon Index in mind. Try to keep these species abundance curves in mind. They will help you make mental comparisons. Okay, we're done for the day.